Good afternoon. I'm George Daley, the Dean of Harvard Medical School, and on behalf of the faculty co-leads Arlene Sharp and Bruce Walker and the entire steering committee of the Massachusetts Consortium on Pathogen Readiness, thank you for joining us today for this conversation on new viral variants and what they mean for our efforts to contain the spread of SARS-CoV-2, to rein in its lethality, and ultimately to end this pandemic. We are just shy of the one-year anniversary of the World Health Organization's declaring COVID-19 a global pandemic. Worldwide, we've seen more than 112 million cases, 2.5 million deaths. In the US, we've seen 28 million cases, and just this week, we surpassed the grim milestone of 500,000 deaths. The toll in lives has been extraordinary, and the economic loss also staggering. There's been tremendous hope, however, injected into this current maelstrom with the most rapid development of vaccines in human history. It was precisely 11 months from the release of the genetic sequence of the SARS-CoV-2 virus on January 11th, that is, to the emergency use authorization of the Pfizer-BioNTech vaccine on December 11th. The Moderna vaccine received EUA soon thereafter, and we believe we are just days away from EUA here in the US of the third vaccine manufactured by Johnson & Johnson. Now, despite some stuttering, the vaccine rollout is well underway. While we in the medical community are guardedly hopeful and optimistic that the vaccines promise the end of the current pandemic, there is cause for concern that with the appearance of viral variants across the globe, we might be facing a decidedly novel stage of the contagion, COVID 2.0. Now, a virus exists to survive, multiply, and spread. Mutations, molecular changes to the viral genetic code, occur every time a virus copies itself. Mutations are the microbiological equivalent of copy and paste errors. Now, these changes occur randomly and in direct proportion to the amount of virus that circulates. More virus means more mutations. Now, most mutations are inconsequential. Some can be harmful to the virus, and yet some can make the virus more fit, more contagious, while some allow the virus to evade the body's immune system. These mutations will, in fact, allow the virus to survive, multiply further, and spread. These are the mutations we worry about. So the emergence of the new variants in and of itself is not surprising. We expected it. But what has been unsettling is how many times these new variants have cropped up with the same mutations independently in infected patients across the globe, and even the same ones in petri dishes in the laboratory. The rise of these variants is the consequence of natural selection, Darwinian evolution occurring in real time. Now, fortunately, experts within our medical community have been paying close attention to the virus as it mutates and stand ready to confront whatever new challenge emerges. The rise of the coronavirus variants forces us to confront a series of critical questions. Are these viruses more dangerous? Will they evade immune defenses? Will they render the current vaccines ineffective or less effective? What do we do next to be sure we're not hit with another pandemic right on top of this one? These are all questions that are central to the mission of MassCPR, the organizer of today's session. These are the questions that fuel the efforts of the virologists, immunologists, epidemiologists, infectious disease specialists, and other experts that make up this multi-institutional consortium that has engaged more than 500 clinicians and scientists from across the Commonwealth. The goal of MassCPR is to address both the immediate implications of the COVID-19 pandemic, but also the long-term need for enhanced preparedness for future emerging pathogens. With that, I will turn it over to our first presenter, Dr. Jeremy Luban, a renowned expert from the University of Massachusetts Medical School, whose work focuses on demystifying viral behavior, pathogen evolution, and immunity. Jeremy, over to you.
Thank you, George. Thanks to you, to the MassCPR leadership for the opportunity that I have today to speak with all of you about these variants of concern in SARS-CoV-2. I'm going to explain to you over the course of this little talk what variants of concern means and how we consider them to be significant for control of the pandemic. Next slide, please. I'm going to start with an interesting historical story about the discovery of human coronaviruses. In World War II, before the US entered the war, James Conant, who was president of Harvard University, established the Harvard Hospital in the south of the UK in Salisbury. And its purpose really was to study infectious diseases that were considered a priority of the British war effort. In 1946, the hospital was converted to the Common Cold Research Unit. It typically would bring volunteers in for a 2-week stay. They'd be challenged with an infectious pathogen and they'd be monitored closely. And from these kinds of studies, we've learned much of what we know today about the viruses that cause respiratory infections and what we can expect when these infections occur. In 1964, David Terrell, who was the director of the Institute at that time, had some samples they were unable to propagate in the lab, and he sent them to an expert in London, June Almeida, who developed many of the techniques that we use today to visualize, to actually look directly at virus under the electron microscope. And she saw these very strange shaped viruses with a halo of spikes around them that led her and David Terrell to christen them a new class of virus that they call coronaviruses. This exact image we're looking at here is a virus that all of us have encountered in our lives. Most of us have been infected by the time we're adults. This virus causes a common cold and raises interesting questions about what will ultimately happen with SARS-CoV-2. Next slide, please. So as George reminded us, in January 2020, SARS-CoV-2 was discovered. It's incredible how fast this happened when we think about the history of virology and how long it usually takes to figure these things out. The virus is a coronavirus. It has the typical spikes around the outside of the virus, and it has an RNA genome that's one of the biggest viral RNA genomes that we know of. It infects cells in the nose and the lung because it binds to a protein on the surface of these cells called ACE2. And when the virus enters these cells, it damages the tissue and causes the pneumonia that we've become familiar with. Next slide, please. So for the virus to replicate itself, it's totally dependent on our cells. And this diagram just shows a coronavirus binding to its ACE2 receptor, releasing its genetic material, in this case RNA, into the cell. The virus delivers machinery that's required to replicate this RNA, to make many copies of it. The RNA as well encodes proteins that form new virus particles that leave the cell and are then capable, in completion of this life cycle, capable of infecting new cells or spreading through the air to new victims. Next slide, please. So with these variants of concern, what we worry about are the appearance of mutations. And it's, as George mentioned, mutations in effect occur randomly. They're like typos in the genetic sequence that are not intended in any way. They're just errors that appear randomly. And sometimes these errors are actually transmitted in the virus. If they're compatible with the full life cycle of the virus can replicate and bring these new mutations in the genome forward in the population. Next slide, please. So one of the amazing things about the COVID-19 story over the past year is that there are virologists all around the planet who have been sequencing the genomes of the viruses that are infecting people. So we have tens of thousands of sequences, each of which gives us a fingerprint of the virus that's infecting that person or that community. This orange virus here is an example of a mutation that doesn't propagate over time. It's perhaps defective or it has no advantage. This yellow virus increases in frequency. And when we see something like this happening, that is a variant virus that increases in frequency, this is suggestive that this virus is more contagious or has some ability to replicate faster so that it takes over the outbreak. Next slide, please. So how might a mutation make the virus replicate more quickly? This is just an example of what might happen when mutations occur in the gene encoding the spike protein shown here. Most of the mutations will be neutral. They'll have no effect on the function of the spike protein. Many of them will be detrimental and will just disrupt the ability of the protein to function, and we'll never hear from them again, even though we may see them once in a sequence from a virus population. Very rarely, maybe 1 in 1,000 of these mutations will have some kind of advantage. Perhaps they'll allow the spike protein to bind tighter, shown by these red lines, to the receptor, allowing the virus to replicate faster. Next slide. Next, please. And one of the concerns that we have is that these mutations in the spike may permit the virus to escape from the immune control that's been established in a person from a prior infection or perhaps from a vaccine. So antibodies, which are, again, just one of many components of the immune system, are thought to function by blocking the spike protein's ability to bind to the receptor. We have seen over the course of the outbreak, that there are mutations which change the spike such that the antibodies that had been previously formed are no longer able to bind to the mutant protein and no longer block the virus at this stage. Next slide, please. So the variants of concern is sort of a general term that we've adopted as a useful terminology. It reflects the sudden appearance of a large cluster of mutations all of a sudden. So for example, these creatures that we've heard about in the media over the past couple of months, like B117, they have these awful names that I have to keep notes, a kind of cheat sheet to keep track of them myself. B117 had many new mutations all at once in the order of 27 new mutations. And that's generally the case with all of these. So we see them appear, we see them take over the outbreaks. And the big questions that come up that are hard to answer often in the real world are, does it actually enhance transmission? Does it permit reinfection in a person who was previously infected? And most importantly, on many of our minds, does it decrease vaccine efficacy? Next slide, please. So I'm going to show you one vignette. It's not a completely understood situation, but it raises a kind of alarm about the kinds of things that we need to think about. Manaus, Brazil is a city in Amazonas where there was a huge outbreak in the first wave. Many, many people died. And a recent paper calculated that up to 70% of the population had been infected and achieved what we would consider a level of herd immunity that would prevent new infection. What was particularly disturbing recently is that there was a huge explosion of hospitalizations in December. Many, many people died. And this corresponded with the appearance of a new variant called P.1, which has many mutations that raise concern about whether the virus is resistant to the immune response against the first virus. Next slide, please. So going forward, it's critical that we maintain public health measures to minimize the chance for new mutations to be elicited by the virus. We need much better international monitoring. The virus doesn't care about borders. We need to sequence, for example, much better all throughout the United States to know what viruses are actually circulating there. And we may very well need to modify vaccines as we go forward to accommodate the mutations. Galit Alter is going to tell us about the vaccines in more detail. And the last point that I think we really must emphasize that this outbreak has pointed out again is that we really need universal access to healthcare and education, because if anyone is left out, they put the rest of us at risk. Thank you for listening, and I'll hand it over to Galit.
Thanks, Jeremy. George mentioned at the start that one of the big questions that has emerged since we learned about the variants of concern is whether these new variants will impact vaccine efficacy. Next slide. So as Jeremy mentioned, our body fights the virus by generating antibodies that bind to the spike protein. Next slide. These antibodies protect us from disease by preventing spike attachment to the human ACE2 receptor on respiratory cells. Next slide. But as Jeremy mentioned, some of these mutations, including the B.1.351 variant that was first identified in South Africa and the P.1 variant first identified in Brazil, occur where antibodies bind to the spike protein. Now, these mutations can therefore potentially prevent antibody binding and potentially compromise the ability of our antibodies to block infection. So the big question that we've been asking ourselves for the last few weeks is whether our current vaccine-induced antibodies can still block the new variants. Next slide. Now, importantly, following infection or vaccination, we make millions, if not trillions, of antibodies that attack the viral spike at different sites with different binding properties. We never make a single antibody to a single site on the actual virus. Next slide. So the big question is whether the emerging variants knock out a few of our antibodies or all of our antibodies induced by vaccines. And the bigger question is whether those vaccine-induced antibodies that still are able to bind to the virus can still provide enough protection against these emerging variants of concern. Next slide. So emerging data has really been coming online literally every minute since these variants have been described. And what I'm showing you here in this particular graph is a comparison of the ability of vaccine-induced antibodies to block different variant infections. So shown on the left column, we're looking at the blocking activity of antibodies to the original strain of the virus. And the two right columns show some level of resistance of the new viruses to vaccine-induced antibody blockade, with a small reduction in the B.1.1.7 in the middle, and then a little bit of a larger resistance by the B.1.351 variant. But what is important to notice on this particular graph, which is really an illustration of all the data that's come out in the last few weeks, is that some blocking activity is always retained in most of the vaccinated individuals, suggesting that we are not losing all of our binding antibodies to this virus, and we retain some level of activity in the ability of these antibodies to see and interfere with viral infection. So the question is that sufficient to bring us out of the pandemic? Now, if we move to the next slide, one of the key missing pieces of information is that our immune system actually fights infections with more than just antibodies. In fact, beyond antibodies that are shown on the left side, during an infection, we also generate a second line of defense, which is shown on the right. We also generate T cells. Now, T cells, unlike antibodies, play a different role in the elimination of viral infection. What these cells do is they recognize infected cells, after which they eliminate these cells rapidly, clearing our body of all evidence of the infection. Now, while the emerging variants seem to affect vaccine-induced antibody blocking activity shown in the little letters on the bottom of the left side of the slide, what we're seeing is that these variants don't seem to affect T cell immunity all that much, and they seem to be as effective in recognizing these variants as they do in the original virus. So what that means is that we actually have very important backup mechanisms built into our vaccines that may continue to provide protection against these newly emerging variants. Next slide. But the best proof that our vaccines still work is shown in the wake of this emergence of all these viral variants is illustrated again on this summary slide. What I'm showing you here is the level of efficacy or protection that we are seeing in recently completed phase 2 and phase 3 trials that are done in regions of the world where the variants are emerging. Now, while we see the greatest level of vaccine protection from severe disease and death in the US in the far left column, where the variants are still rare, we do see a stepwise decrease in vaccine efficacy in parts of the world where the variants are more abundant, like the UK and South Africa. But the good news is that we are still seeing significant levels of protection in the face of the variants that are evolving, again, begging the question of whether this level of protection will be enough to get us out of the pandemic. So if we go to the next slide, the key to understanding if these responses are sufficient to get us out of this pandemic is summarized on this slide and relates to the mechanism by which vaccines prevent disease. Now, what we have learned over the past year since the SARS-CoV-2 pandemic began is that the virus itself causes largely mild disease, shown as little orange people. And that occurs in most of the individuals that come in contact with the virus. But unpredictably, in some individuals that are vulnerable, the disease can cause, or more severe disease can occur. And we've depicted this in the top middle graphic as little red individuals. Now, on the bottom left cartoon, without a vaccine, masks, or social distancing, what we know would happen is the virus would spread relentlessly, causing disease and death. However, what we've seen from the phase 3 studies in the bottom middle cartoon is that in almost all the vaccines that we have tested so far, the vaccines are remarkably effective in limiting the number of individuals that develop severe disease or unfortunately pass away. So we are able to eliminate the conversion of individuals into those red, highly sick individuals. So what that tells us is that the current vaccine portfolio is able to essentially turn COVID-19 into a common cold. However, there's been some really interesting data in the media emerging from the UK and Israel, shown or depicted on the bottom right side of the graph, that suggests that some of the vaccines may also prevent transmission, essentially blocking both the development of severe disease, but also completely preventing individuals from getting the infection at all. So no mild infection either otherwise. And that happens by blocking the virus completely from entering into the human population. Now, the problem with that bottom right graphic is that the big concern is that antibodies that are required to completely block transmission may be more affected by the new variants. But let's just assume that if we just focus in on that bottom middle graph, even if we can't block transmission with the vaccines that we have in our current portfolio, if the current vaccines can turn COVID-19 into a common cold across the globe, like in that bottom middle graph, these vaccines will surely help us get out of the pandemic. Now, if we move to the next slide, the essence to really understanding if that middle model can actually work really depends on understanding how these vaccines can provide population level protection, otherwise known as herd immunity. Now, the way that herd immunity works is depicted here. On the top line, where we see no vaccination, in the absence of a vaccine, an infected person, shown in the red, is able to spread the virus to many non-immune individuals in the blue, in the middle, and then the red takes over all the way on the right. However, with a highly protective vaccine, blue individuals can be rapidly converted into green immune individuals that limit the spread of the virus, so very few people essentially turn red. But even in the bottom, with a moderately protective vaccine, even if we go down from 95% to 70%, what we see is that immunity conferred by the vaccine can essentially completely limit the breakout of any types of infections in a population, really showing us that even at lower levels of efficacy, this may be sufficient to prevent population level spread of the infection. This data gives us hope that even with the vaccines that do not confer 95% protection, even against these emerging variants, the light at the end of the tunnel is approaching. Now, if we go to the next slide, the one question that also keeps popping up is whether or not this is going to be a yearly vaccine, the COVID vaccine, or is it going to be a vaccine that is going to be administered once in our life? Now, unlike flu, shown on the left side, that rearranges in really fantastic ways, both through genomic reassortment and individual mutations that require a yearly vaccine, it is possible that the SARS-CoV-2 virus may only require a single vaccine that we may evolve over the next few years as we learn about the variants of concern. If we move to the next slide, what is happening right now at the level of the vaccine development is that we are moving very quickly in identifying the variants that are able to escape the current vaccine-induced immune responses. And so what is being developed as we speak are next generation vaccines that may drive immunity to potentially critical variants shown on the top right side, or to potential combinations of variants that may allow us to develop highly flexible responses that can essentially get us out of this particular pandemic and prevent new pandemics from occurring. Next slide, and one more. And so what I'll leave you with is this idea that essentially where we are today is really in an unprecedented place where we have more vaccines for this virus than ever before. We are learning about these variants of concerns, and our current vaccines do provide the level of efficacy that we believe will bring us out of this pandemic. But these new variants are inspiring novel vaccine development and design that will surely lead to the development of vaccines that will help us move past any future pandemics to come. And with that, I will turn the mic over to Jonathan Abraham, who will tell us about the concerns with these variants on emerging therapeutics. Next slide.
Let me just interrupt quickly and give a brief introduction to who Galit is, and then I'll turn it over to Jonathan. Galit is a professor of medicine at Harvard Medical School and the Ragon Institute. She's an esteemed immunologist who co-leads the pathogenesis working group of MassCPR, and she has dedicated the past year of her life to unraveling these fundamental mechanisms of how SARS-CoV-2 interacts with our host to induce immunity. So thank you, Galit, for that presentation. And now our next presenter is Jonathan Abraham, who is an infectious disease specialist and a physician scientist, assistant professor of microbiology at Harvard Medical School. He has indeed made the study of emerging pathogens his life's mission. And that was long before the COVID-19 outbreak. And Jonathan co-leads the Therapeutics working group of MassCPR. Jonathan.
Thank you, George. So the title of my presentation is Staying Ahead of Viral Mutations with Therapeutic Antibodies and Small Molecule Drugs. Next slide, please. And so here what I'm showing you is a schematic diagram of the SARS-CoV-2 viral particle. As you can see, it's decorated with these club-shaped molecules here shown in red. As Jeremy and Galit mentioned, these are the viral spike protein. The point I'd like to add is, of course, when we're infected with SARS-CoV-2 and recover, we have these Y-shaped molecules that circulate in our blood and can bind and inactivate the viral spike protein. These antibodies could also be elicited by vaccination, including with the current vaccines that folks are starting to get at this point. But most excitingly, they could also be developed as drugs, and that's what I'll tell you about in the next slide. And so in this case, the way these antibodies are found mostly is you could take somebody who has recovered from SARS-CoV-2 in this case, and they will have in their blood these antibodies shown in red and green here that have particularly favorable properties, like the ability to efficiently inactivate the virus. And so one can take an aliquot of the individual's blood, in that blood, get the immune cells called B cells that make the antibodies and recover from them the genetic sequence to produce more antibody. So in this case, for example, I'm showing you in the saline bag what would be a monoclonal antibody cocktail of two antibodies, a green one and a red one, that would have activity against the virus. So these can be produced in large quantities, again, infused into saline bags that could be administered to uninfected patients as a way to confer them with instant immunity. It could also be used in individuals who are perhaps in the early stages of infection as a modality to, again, treat them. Next slide, please. And so the molecular mechanism, I think Galit and Jeremy mentioned, but effectively the antibodies will bind the spike protein and prevent it from attaching to ACE2, which is the cell surface receptor. So this is the first point of contact of the virus with cells. The virus actually can bind ACE2 and perhaps still sneak in, but a second class of antibodies can lock the spike in the conformation that inactivates it, so it's unable to allow the genetic content of the virus to be released into the cell. And so most of what we know about antibodies to the spike protein, again, target this receptor binding domain, which is shown on the right part of the slide here in blue. This part of the protein is the furthest away from the membrane of the virus, which is shown at the bottom of the slide, and is an attractive target for therapeutic antibodies. Next slide, please. And so multiple monoclonal antibodies that bind this receptor binding domain are now approved for emergency use in humans, and others are being developed. For example, Regeneron has a monoclonal antibody cocktail I'll mention in the next couple of slides, as well as Eli Lilly with a monoclonal antibody that binds specifically to the RBD. These antibodies will bind and block ACE2 engagement. So most of the antibodies that are being explored as therapeutics function through the same mechanism. And perhaps what's been the most daunting is that previously predicted antibody resistance mutations detected in the lab are now being detected in variants circulating across the globe, meaning that the virus is sort of behaving in predictable ways. Next slide. And what I mean by predictable ways here is if you look at the left part of the slide here, I'm showing you what the receptor binding domain in blue looked like in, say, December of 2019, January of 2020. With the B117 variant, we see on the RBD a single mutation here that is exactly where the antibodies would bind. And then with the variant first detected in South Africa, the B1351, you'll see that there's 3 of these red spots on the spike protein RBD. And then I mentioned where we're probably headed from the standpoint of, again, these predictable mutations. There's room for probably at least 8 or more mutations in the antibody footprint that would effectively perhaps limit their effectiveness in the long term. Next slide, please. And so most monoclonal antibodies you would predict should be active against the B117 variant. As I mentioned, there's a single mutation on the RBD. The RBD landscape, however, is rapidly changing, as I mentioned. And then multiple antibodies targeting different parts of the spike are probably what is needed to counter resistance. And I'm giving you an example here of the 2 monoclonal antibodies from the Regeneron cocktail, Regeneron 10987 and 10933. And as you can see here, the spots on the RBD that are changing, again shown in red, are the spots exactly where the antibodies are binding. Next slide, please. And that being said, the good news is that there are other viral proteins that can serve as drug targets. As Jeremy mentioned, SARS-CoV-2 has a very, very large genome. It requires a lot of other proteins to infect cells and spread, about 30 or so proteins. And some of these proteins are enzymes, and enzymes are what the pharmaceutical industry has tons of experience targeting with small molecules, which can potently inactivate these. So I'll give you an example here of remdesivir, which is an FDA-approved drug now for SARS-CoV-2. And so remdesivir works by blocking the viral polymerase. This is the part of the virus that is effectively making copies of the viral genome, so effectively a copying machine that can be blocked by this inhibitor. Now, the challenge that remdesivir has to be administered intravenously, so by infusion, but now there are new classes of drugs that can be administered as pills by mouth that are causing a lot of excitement in the field as potential therapies for SARS-CoV-2. Next slide, please. And so in terms of thinking about how we can prevent future outbreaks, as well as effectively treat variants of concern, where, again, most of the changes are happening elsewhere on the spike protein, virus enzymes typically mutate much less frequently than the spike protein. And so small molecules targeting them are attractive strategies to counter variants, as well as have activity against multiple coronaviruses. Here I'm showing you on the left side part of the slide the crystal structure of the SARS-CoV-2 protease. This is an enzyme that's required to effectively cut and activate some of the viral proteins for them to be functional. And so the main protease's crystal structure is a perfect example of whereby scientists could use this structure, identify pockets that would be well suited for binding by these small molecule inhibitors. But you can imagine that that kind of drug discovery happens on a much longer time frame. And so while small molecule drugs are attractive as pan-coronavirus drugs, they typically take a much longer time to make. Next slide. And so conclusions here would be that spike mutations, as they continue to accumulate, will impact monoclonal antibody drugs. New antibody combinations or cocktails must target multiple parts of the spike protein that are less prone to mutations. As mentioned as well in some of the questions that are coming in, there are other mutants being described throughout the globe. And so probably with the B117 and B1351, as well as the P1 variants being described, there are additional mutations we really have to be on the lookout for through vigorous genomic surveillance. Small molecule drugs can work to target other proteins that are not mutating as fast, but again, these take longer to develop. And a critical question that remains unanswered, and I say is the elephant in the room, is really for drugs to work against SARS-CoV-2, how much time do we have to give them after somebody gets sick to prevent them from getting sicker? So we know from influenza virus, for example, that drugs typically have to be given within the first 48 hours to really have an impact. And so that same time window is not clearly well defined for SARS-CoV-2. And so it's a critical question to answer is effectively when to use the drugs, how quickly. So on that note, I'll wrap things up and pass it on.
Thank you very much, Jonathan. And thanks to the speakers. Those were very, very tight presentations, covered an enormous amount of information. But there are still quite a few questions, and I would encourage anyone listening, if you are interested in asking a question, please use the Q&A feature on the Zoom platform. And I want to say that in addition to Jeremy, Galit, and Jonathan, we're being joined in this question and answer session by the faculty co-leads of MassCPR, Drs. Arlene Sharp and Bruce Walker, as well as Dr. Mark Namchuk, who together with Jonathan Abraham co-leads the Therapeutics Working Group. As well, we have Lindsay Baden, an infectious disease physician and the anchor of the phase 3 Moderna trial, and also the infectious disease editor of the New England Journal of Medicine. And then finally, Dr. Dan Baruch, whose work in the laboratory generated the adeno-26 viral vector that is the foundation of the Johnson & Johnson vaccine. So it's our privilege to be joined by such a distinguished panel of speakers. Let me take the first question and ask Jeremy perhaps to spend a bit more time talking about what we know about the California variant, which has very much been in the news in the last 24 hours.
So the California variant has been on the radar of the virus community for a couple of weeks now. It's clearly increasing in frequency and therefore of concern. As I mentioned in my talk, it's well, that's sort of the initial signature, the increased frequency of the variant is the initial signature of something being more transmissible and being of concern to us. It doesn't necessarily mean that it is more transmissible. One of the difficulties assessing that particular mutant is that it appeared at a time when there was a surge in infections. And if we look back to the kinds of studies that were necessary to demonstrate clearly that B117 was indeed more infectious in the UK, where we had much more data, logs more data, many more people in different contexts, it took quite some time to prove that it was more infectious. So most of us believe it's going to be more transmissible, more infectious from what we can see, but that's not clear. It may have just passed through a bottleneck and be the creature that happened to be there at that particular time. As far as the lab data with it, it looks like it's very modestly more infectious, but quite modest. And the little bit of neutralization data that's been put out in the public so far is pretty mild in terms of the effects of the mutation. There are claims that it's much more pathogenic, that people get sicker, but I think that those are pretty unlikely. The data is very, very small at this point, and effects like that really would be very surprising.
Great, thank you, Jeremy. There was a couple of questions that hinted at the multiple arms of vaccine-induced immune responses. Galit spoke to it to some extent. I would ask Bruce Walker, who's an expert in this area, perhaps to expand on strategies for goosing the T cell arm of the immune response. Bruce.
Thanks, George. Yeah, I think this is something that has promise for turning, hopefully, this family of coronaviruses into something more like the common cold. T cells target not just the spike protein, which is so variable, but also other parts of the virus that really cannot change and leave the virus still able to be as virulent, to be able to replicate as well. And so one of the vaccines or two of the vaccines that are currently in use or being tested, the Oxford vaccine and Dan Baruch's vaccine with Johnson & Johnson are based on an adenovirus, and those do induce a level of T cell response that's higher than what one sees with the mRNA vaccine. So I think this is an area where there is an opportunity to broaden the response. And I would point out that for influenza, it's likely that year to year, part of the protection or part of what makes the virus less severe is the presence of cross-reactive T cell responses. I don't know if Dan wants to add anything to that related to the J&J vaccine.
Dan?
Sure. So I think in light of the speakers that we just heard, it's very clear that multiple aspects of the immune response are needed. Neutralizing antibodies we've heard about the most for SARS-CoV-2 vaccines, but other functional antibodies that are not neutralizing are also likely very important, as well as T cell responses. So just like you wouldn't want to fight with one hand behind your back, I think for this virus, we really want all aspects of the immune response triggered by vaccines. And I think that's particularly important for viral variants that might knock out or severely reduce one or another type of immune response. Then it's likely that other types of immune responses can kick in.
Sorry, I was on mute. There's a question about the impact of vaccination on the ability to not just reduce disease severity, but can vaccination, in fact, prevent people from getting infected at all so that we can be assured they're not carrying the virus? There's been concern and certainly recommendation that even vaccinated individuals should maintain mask wearing and social distancing because there's still the risk of some carriage and transmission of the virus, even if they themselves are not symptomatic. Could perhaps I ask Lindsay to comment on the emerging data that is, I think, giving us an encouraging sense.
So George, thank you. I mean, it's so important to understand carriage and transmissibility. The studies that were the basis for regulatory action looked at illness with an eye to severe illness, which is the appropriate yardstick for primary approval and best clinical benefit. But the question of transmissibility is so important. And I think Bruce alluded to the issue of, can we turn this into the common cold? And different aspects of the immune system can ameliorate the illness so that one doesn't feel sick and perhaps even doesn't acquire the organism. And that is the goal. But we don't yet have the data. And so the data are being generated. There's a hint from some of the reports from the UK and elsewhere, and even the early data in the phase 3 trials, where there is decreased carriage of SARS-CoV-2 by PCR in the nasopharynx in vaccinated individuals, suggesting that there will be a decrease in acquisition and a decrease in transmissibility. But those data, we require larger data sets. and to actually look at viable virus and transmissibility, because nasal carriage in and of itself doesn't mean transmissibility. So I think we will have more data shortly, probably a month or two from now. There'll be more data that directly speak to this point, but the emerging data are very encouraging that nasal carriage is decreased in appropriately vaccinated individuals.
Thank you, Lindsay. So we really do have some positive messages here. I think we're hearing that even though we hear lots of scary news reports that the new variants may be increasingly transmissible, they may be more virulent and cause more death and may evade the immune system, there seems to be a real cushion, as we're hearing, that there may be some modest reduction in the laboratory measured immune response, but there seems to be protection, and let's maintain some optimism here until the data tells us otherwise. A quick question for the clinician. I've got Jonathan and Lindsay. Patients with autoimmune disease who are maybe under immunosuppression, are they at greater risk for the variants, and should they be vaccinated? Jonathan, perhaps?
Oh, yes, I can start. I mean, that's a very difficult question. I think from the standpoint of the new variants, what we know thus far is that, at least from the data I've seen, none are particularly concerning from the standpoint of being more lethal. It's more of a question of the degree to which the spike RBD can further evolve in those situations. So, and experiences I've seen, those individuals, particularly conditions, might not necessarily be sicker, but just carry the virus much longer in their system. And with that, persistent infection can eventually develop additional mutations that then could have an impact on, effectively the virus spreading. So I would say that in those situations, perhaps the best strategy would be to use monoclonal antibodies that do work and use them early to prevent further viral evolution.
Thank you. There's a question about the next generation vaccines, and I might ask Dan, are there plans underway in the Johnson & Johnson vaccine program to develop new versions that work against the variant, and are those efforts underway in Moderna and Pfizer as well?
Yes, I believe that all of the major vaccine manufacturers have announced that they are making updated, revised vaccines that include the B1351 variant that originated in South Africa. That includes Moderna, Pfizer, J&J, Novavax, and AstraZeneca. So I believe that all 5 of the major vaccine developers in the Western world are doing that already. I think the extent to which they are going to be needed, and if so, how they are going to be implemented, remains to be determined. The recent data showing good protection against the variants is positive news, but it is somewhat reduced compared with against the original Washington strain. So how that will play out in actual populations in the United States, in Latin America, in South Africa, in Europe, is not yet known. But I think the prudent thing to do is to prepare revised vaccines. And if they're not needed, that's great. And if they are needed, we'll have them.
Dr. Baden, are we going to have to wait another 4 or 5 months and enroll 30,000 individuals to have these new generation vaccines approved, or will there be a more streamlined strategy?
So how do we make the new vaccines available if needed? And that's a regulatory question, and very likely correlates of protection will emerge, which is in the vaccine studies we've done, do we understand what correlates with the protection of those individuals who didn't get sick versus those who did? So vaccine failures. Once we can help establish those correlates, then immunobridging is very likely to be the way for new vaccines to emerge. And that's an ongoing discussion with regulatory agencies and with the scientific community. But I think it's highly likely that that will occur.
Great, thank you. Very interesting question. I will target to Galit and Arlene as our immunologists. There's a question whether there's any likely differential effect of the variants on children. Children seem to be less vulnerable to the severe acute respiratory syndrome, but have this multi-system inflammatory syndrome. Anything known about the pediatric immune response that is informative?
Arlene, do you want to start?
Why don't you start?
Okay. So there's no data yet, George, to say that these variants will have cause more pathology in children. I think that the one concern, though, is that we see that these cases of this multi-inflammatory severe disease always occurs a month after we have large waves of infections happening in populations. And so what the example that Jeremy showed with Manaus, the scary kind of picture there, where you get this rapid emergence of a single variant, emerging in that population, means that a month later, you're also going to have a wave of kids coming in with this really unusual Kawasaki-like disease. So I think that what the hospitals are doing is they're essentially bracing themselves for the possibility that these kids are going to come in, and they're trying to prepare more effective treatment, protocols to make sure they can provide the supportive care that is required to these kids, because as of yet, we still do not fully understand what it is about this particular infection that drives this unusual phenotype in these kids. And it always happens that 1 month after the virus has spread through the adult population.
I would add to that. That's great. That the timing here is really something we don't understand why it is happening. And so I think this provides an opportunity, as we're prepared now to know the timing of this, that we can then do more to understand why this is happening, and that'll help us come up with better therapeutic approaches.
Great. Well, let's talk a bit about drugs that aren't antibodies, but are direct antivirals, which have proven so powerful against HIV and other viruses, and there's great hope for coronavirus. Mark Namchuk runs the Therapeutics Initiative at Harvard Medical School and is the co-lead of the Therapeutics Working Group. There is a question about whether there are other strategies, such as blocking recombination as a strategy. Perhaps you could speak about the array of targets and what the timeline might be for delivering a drug.
Sure. Thank you, George. I think I'll maybe go back into two parts here. I think using HIV as a template, one of the most effective strategies has been, as Jonathan Abraham introduced earlier on, to really focus on enzymes as targets that are directly encoded by the virus and directly involved in replication of the virus. So to use an analogy, the spike protein's a little like the hood of your car. It can take a whole bunch of dents and the car just works fine. The enzymes within the car are really the engine and the mechanism by which that car moves, and they're required for viral replication. So I think the two targets that are getting the most attention are the polymerase and the main protease. I think you're seeing advances on both fronts. There is at least 6 molecules I'm aware of that are now targeting the polymerase, and at least 3 that are targeting main protease that are in earlier stages of development. In terms of going after recombination, directly to the question that was asked, I haven't seen much emerging on that front for new molecules so much. I think there's been a large investment in looking at different host cell factors that may be involved in driving replication along and parallel lines to what we're thinking about with the direct viral proteins. And there's some of the clinical results to date from repurposing haven't been quite as successful as targeting the virus. Lastly, in terms of timeline, the remdesivir molecule, to use it as a template, already had phase 2 experience clinically. And so it was able to rapidly move towards an approval in a phase 3 trial. A more normal timeline from the point that you have something ready for first in human dosing through approval would probably be on the order of a few years, 3 to 5 years at minimum. And I think that's maybe the last comment I'll make to what Jonathan was saying. Investing in making sure those molecules might be able to suppress, to still be effective with viral variants that emerge within SARS-CoV-2, but also as our dry powder, so to speak, for another coronavirus, and ensuring they have spectrum. And having that as a first line of defense, I think, is an important aspect of things that we should think about as for future therapeutics.
Perfect. Thank you. There's a couple of questions about viral sequencing and genomic surveillance. We've heard in the news that other countries like the UK seem to have invested more in surveillance strategies and have been able to identify the variants quicker. Are we doing enough in the United States? And if not, why not? And what needs to be done? I might ask Dr. Walker to tackle that one. Anyone else wants to offer that?
Yeah,
I can jump in too.
yeah, I'll just say a short word and then let Jeremy take over. No, the short answer is no, we're not doing enough, and that we need to track these things in real time. I think the other aspect of this is that we really need to follow people with sentinel surveillance who have received the vaccine to see what is happening to those people if they do get infected. What is the level of protection that's being followed? It's being generated, and what are the variants that are getting past that barrier? But Jeremy, why don't you go ahead and speak to that?
So the UK really is remarkable for many reasons, including the fact that they have a national healthcare system that's made it much easier to link any sequences they get to the clinical context to figure out what the significance of the sequences are. It's also not really fair to compare the US to the UK, in that the UK is sort of the equivalent of a big California, and we have 50 states, each of which has its own regulations and its own public health departments. And a big issue in the United States is having a kind of central coordination that links all of the country together. All of the regulations are big obstacles. There is a very rapidly increasing rate of sequencing occurring in the US. The CDC is starting to take the lead on that, and we're starting to get information. But the UK sequences about 10% of all of the viruses that infect people, and we're still at about the 0.4, 0.3%. So we have a long way to go. The significance of this is that we do not know what's out there. And without that knowledge, we can't plan, we can't design vaccines, we can't know if there's something lurking that we need to worry about.
And Jeremy, what level of sequencing do you think is appropriate, is needed?
Yeah, so the epidemiologists tell me that we really should be in the 10% range, or certainly 5%. Also, not just the number, but we need it distributed. So there are big parts of the country where there is no sequence information, and it's really fallen on the shoulders of individual academic labs in specific locations to do this. And that's really what we've seen in the US, kind of to put it kindly, a mosaic of information that really needs to be more of a coordinated effort.
Well, thank you. I'm sad to say we have come to the end of our hour, and there's a growing number of truly provocative and interesting questions. I'm sorry we won't have time to get to, but we will continue these public briefings. I want to thank the speakers. I want to thank the members of our panel for offering their expertise. One of the questions that I want to leave everyone with is, how do we communicate in this evolving time with so much uncertainty? I think what you've heard is a strong message that despite the emergence of the variants, we do believe that the current array of vaccines will prove effective. And if they don't, we will cross that bridge, and we are ready to confront whatever comes our way. We have strategies for evolving the vaccines into the next generation to be effective against variants, and we have impending efforts at drugs. So I remain hopeful, and I hope all of you as well. But in the meantime, I remind everyone that we have months to go before we achieve herd immunity. And during that time, whether you're vaccinated or not, please do your part in adhering to the public health measures of wearing your mask and remaining vigilant. We hope sometime in the not too distant future, we will be able to tell an even more optimistic story. So thank you very much. Be well, be safe, and be healthy. Until next time.